Hello, hello, and welcome to this poster presentation. I'm Carolin, and I'm going to talk about clusterExplorer, an R-Shiny app for single-cell RNA-seq cluster visualizations. So first, a little background. clusterExplorer is a tool for the analysis of single-cell RNA-seq data. From a bioinformatics point of view, it's a biological technique that offers insight into cellular processes. It's a complex technique, and the resulting data is complex too. So we are usually using clusterings, let's say UMAP or t-SNE-based, to find similar cell structures or cells. The example data below is some cancer data, some ependymoma data. This is just an example for clusters and the structures here, so it's not relevant which cancer type it is. So let's say you have this kind of full set with some cancer samples that you are interested in, and you have a subgroup here, and then you can see that clusterings will serve different purposes. You can use different resolutions, and perhaps you're interested in some finer substructures in the subgroup, but you would use an overview clustering of the full set. And then you can just, you may be interested in finding these subgroup structures in the full data set. Let's say you have a trajectory or something, and then you may really be interested in finding some relapse specific smaller structures in the full data set with a whole tumor group. clusterExplorer offers visualizations options for precisely that. So for tracing these kind of substructures in the bigger data set. And yeah, as I said, clusterExplorer is an R-Shiny app. It's built on a pre-processing step with Cell Ranger, Surat, and R. And the main app is based on a pairwise visualization of a chosen set, let's say the big tumor set, and a subset, let's say a relapse data set. And yeah, here the relapse or the smaller data set is on the left side. You can pick any cluster that you're interested in. And this cluster is visualized in red. And it's now projected or traced into this bigger data set, and also depicted in red here on the right side. You can already see that this, let's say, more compact cluster on the left is also quite visible on the right. It's just a bit more scattered, and there are some outliers here on the upper side of the plot. The second option that clusterExplorer offers is a convex hull-based visualization. There you can also see a bit more of the structure, let's say whether or not these cells are concentrated in the middle or on the outside, because there are different lines or circles here, or almost circles, for 25, 50, 75, and 100% of the data. So you can basically see in the middle here, this cluster 3, this darker blue. It's quite compact in this native clustering. And there are some outlying cells in the projected cluster. So you can see that the outer line here, the outer convex hull of the 100% of the data, is a bit larger here. We have also some statistics. They are based on the overlap of these cells with a projected or with a full cluster clusterings. So you can see that if you project these clusters 3 into the full data set, you get not a perfect match, but you get cluster 0 with 60% overlap of these cells, and then almost 30% for this cluster 7. clusterExplorer also offers a full cluster view where you have these convex hulls for all the clusters, and this serves mostly for this kind of structure classification. You can see that there are 2 clusters that are really scattered, this cluster 9 and cluster 6. And that's something that you will have a bit harder time to find in this original cluster view, where you can really, well, you can see it, but you really have to compare the colors. It's more visible here in the clusterExplorer view. For this classification, we use some basic metrics: the distance between pairs of points and the area for quantiles, let's say this 25, 50, 75, and 100% of the data. And you can see here, well, a scattered data set obviously has more space between its points in, well, at least in the mean or median. And the compact structure is a bit more regular. So with a scattered structure, you have this kind of outer line with the 100% of the data. These outliers usually push this kind of bond or this circle a bit more outwards, and this area gets bigger and bigger. And for the compact structure, it's a bit more, well, regular and not so big. So this is 2 metrics which in combination can really discriminate between the compact and the scattered clusters. We are also interested in structures between sets. There we just use a simple overlap, and we call a cluster conserved if it has a, well, if you pick a native cluster in your, let's say, smaller data set and project it into the bigger data set, if you have really a matched cluster. So if most of the cells really land in one cluster, and this would be a conserved clustering then. In case of the example that we have seen, it's more like you need 2 clusters to really find most of the cells of this native clustering. We would call that a split cluster. Or in the more extreme case where you need more clusters, then it would be a distributed cluster. Here it's again the sample. This is cluster 3. And as I said, it's 60% in cluster 0 and 28% in cluster 7. So this is more a case of a split structure. If there were even more clusters necessary, then we would call it a distributed cluster. So thank you for your attention. I'm happy to take questions on the poster and also per email if necessary. And all the references are also presented on the poster. I can say thank you for your attention and see you on Sunday, Monday, or Tuesday. Bye-bye.